So, welcome everyone to day two. We're going to start off with a session talking about some of the regulation around challenge models and try and rehearse a bit some of the differences in different countries. And can I just check that David Caslow is online and can hear us?
Can hear you. Good morning, Andy.
Good morning, David. Great to have you join us. So we'll start with just a very quick round of introductions. So I'm Andy Pollard. I'm director of the Oxford Vaccine Group and have been involved in challenge models for about 15, 16 years now. And we'll go round the table and just ask for brief introductions from each of the panel members. So Esma.
Esper Calas, I'm an ID and currently the director for the Butantan Institute, São Paulo.
Hi, David. Good to see you. I'm Anna Durbin. I think the audience knows me because I spoke yesterday. So.
Daniela Ferreira. Hi, David. Nice to meet you. I'm part of the Oxford Vaccine Group and one of the organizers of this conference.
And David, over to you to introduce yourself.
Sure, David Caslow. I'm the director of the Office of Vaccines Research and Review here at CBER, US FDA.
Great. Well, thanks very much, all of you, for joining us. So the idea for this session is to have some discussion about as I said, about the regulatory aspects of challenge trials in general, but specifically about Zika. And I think it's worth just, first of all, being clear exactly what we're talking about. Because I noticed yesterday that some of the questions and the talks, there's a confusion around the word regulation. So when we talk about regulation in this context, we're talking about the competent authority in a country which has responsibility for quality of manufacturing, looking at clinical trial protocols and the safety and the design of trials, and then also involved in licensure. So it's that regulation around the process of clinical development, manufacturing, and licensing that regulation is about. And whereas yesterday some of the use of the word regulation was about ethics committees, and we're not talking specifically about ethics committees in this session, although I think we may touch on it, and I might ask Anna to comment a little bit on that. But we should be very clear when we're talking about these, whether we're talking about a regulatory viewpoint or an ethical viewpoint, which has a different role. And I think it's worth perhaps just starting by making the point that in different countries, the role of the regulator is very different. So if you go to the UK, for example, there is no regulation of challenge studies. The national regulator, the MHRA, is not involved in challenge studies. And the reason for that is that in law, there is no requirement for challenge agents to be regulated, their manufacture or the use of them in a challenge study. Now, if you do a vaccine, an experimental vaccine, and test it in a challenge study, the regulator will look at the vaccine in the same way as they would in any other clinical trial. But when they look at the challenge part of it, they're looking at it from the perspective of can it be used to measure efficacy, but they're still not looking at the manufacturing of the challenge agent and the use of it beyond its use in that clinical trial. And that means that in the UK system, it's the ethics committee that's responsible for the ethical part, and it's the institution running the study that takes all of the legal responsibility. And that's a very different situation from that in the US. And I wonder, David, whether you'd like to just outline the US system and how that's managed.
Yeah, no, it certainly falls under the purview of the US FDA. And in the case of infectious diseases, for the most part, particularly for vaccine indications, it would fall under CBER and the Office of Vaccines Research and Review. And you're right, I think it's very different here in the United States. Some examples of which is that we typically require that challenge model to be manufactured under good manufacturing practices. We require those trials to be run under a US IND. And so, yeah, I think it's a very different environment here.
And so that process of the FDA regulation means that when you produce the challenge agent, it's usually produced in a GMP facility. And so you know exactly all of the methods of production, and you know what's in the vial at the end. And of course, we do the same in the UK, but it's not regulated, and so there isn't an independent review of that process. But of course, we still essentially follow the same principles. And when you look across Europe, the approach is quite variable, because some countries are very unclear, I think, about how to manage the regulation of challenge studies, and so don't do them at all. And in some countries, they're regulated in the same way as happens in the US, so with very tight scrutiny by the national regulator. So that variability actually makes it quite confusing in Europe. So the EMA, the European Medicines Agency, only gets involved at the point of licensure, because they don't run the clinical trials and the development. That's all run by national regulators, who then have very variable views on how to manage things in Europe. So, Anna, perhaps I could turn to you about your experiences with Zika and regulation. So what is the approach that's taken? How do you make a Zika challenge agent, and then what happens in the process of trial design?
No, I think that's a great question. And when we were developing the Zika Human Challenge, we wanted a natural isolate. And we didn't want a virus that had been recovered and then passaged in tissue culture where mutations could have come in. So we were lucky enough to have investigators here in Brazil and in Nicaragua send us a serum aliquot from a patient who had diagnosed Zika. And that comes with it a little bit additional testing that needs to be done, because then you have to be concerned about human pathogens. And we don't have the medical history of those people from whom the isolate was made. So we have to do additional testing, looking for making sure that, for instance, there was no evidence of HIV infection, hepatitis infection, any infection that could be transmitted along with that virus in our challenge strain. And so Dr. Whitehead had to essentially purify the virus from the serum to try to get as much of a single isolate as he could. And then there was additional testing once that was recovered to ensure there were no adventitial viruses that could have been carried from the isolate. So I think in that case it required additional testing, say, as compared with if you're making a recombinant virus from DNA, you know exactly what the sequence is, you know exactly all of the reagents that you've used to harvest that virus. But when it comes from a human isolate, there's additional testing that needs to be done to ensure that you're not going to transmit any type of infection along with that virus. And then the other, and I'm sure you have this in the UK and others, the other thing that we have to be concerned about from also a regulatory process, and it's a more internal institutional regulatory process, is the biosafety committee. So we have to ensure from our institution that we have measures in place to prevent transmission to the community and that sort of thing. But I will say, you know, working with CBER, and we work with CBER for all of our challenge studies, it, in my opinion, has been a very straightforward approach. We approach it as we would any other investigational new drug. We know the standards that have to be met in terms of manufacture and testing, and we know we work with them to identify any special issues that may come up with that particular pathogen. But because it is working with a regulatory authority, there's guidance all along, and we follow the same guidance for a challenge strain as we do for a vaccine. And I think that's really, in some respects, made it a more streamlined process. It does add an additional step because it has to go through the formal FDA review, but and David can probably speak to this. I think the FDA, particularly CBER, in my experience, is very familiar with these challenge strains now. We did have to work with CDER for a drug study, and it was a very different experience, and I'll leave it at that.
So David, do you want to respond to that?
Well, yeah, I think you make a really good point. I've heard that as well. It really depends on what indication you're seeking or what the intent of that challenge model is as to where it goes within the US FDA. So if it's a vaccine, it'll come to CBER for an infectious disease indication, but if it's a therapeutic, it go to, and you're right, I think there are some differences in approaches between those two centers. I'm glad to hear that the process is at least straightforward, but you're right, it's considered just like it would be any other investigational intervention.
Yeah.
So, Esper, here in Brazil, I know you're not representing Anvisa, but and you're representing Butantan, but from a regulatory perspective, how are challenge studies viewed from a regulatory? Sorry, I need to give you the microphone.
Well, thank you. Thank you for the invitation for this meeting. I'd like to apologize for not being here yesterday, so could make only today. So I missed all the richness, the entire richness of the discussions that took place the first day of this meeting. So before I start saying something, I would like to say that Miruzi Freitas, who was supposed to be here representing Anvisa, could not make it. She had a last-minute problem, but she called me, and she said that Anvisa, the regulator in Brazil, never received a study for challenge up to date. So, but nevertheless, she externed her view that any kind of challenge study that would involve Brazilian investigators and Brazilian volunteers, she manifested that Anvisa is open to start discussion even before the protocol is finalized. And they are willing to contribute to building up the blocks to make a better product, a better project forward. So I'm just passing on the messages she sent. But it gives me the opportunity to also review briefly with you that any study in Brazil that involves human volunteers needs to go through different pathways, and sometimes in parallel, sometimes they need to go in tandem. But we have the ethics committee that scales up to the Central National Ethics Committee. We do have the Anvisa, which I just mentioned, that would be represented by Miruzi here. It's the equivalent to the FDA in the United States and EMA in Europe. And finally, we have the Biosafety Committee that sits in Brasilia, who also reviews any product that involves recombinant pathogens. So anytime that we needed to run a study, even challenge studies here, we would need to go through all these paths to make sure that we have all the approvals in place. And depending on the funding source, you need further reviews. I mean, it's a little bit complicated, but despite being complicated, Brazil have been, understanding this better and better. The intricacies of these agencies have been more clear to us, and we can navigate that and bring studies to the country. However, we did not have any study, the human challenge studies conducted in the country so far. I mean, it's just, it's within our radar. There were some attempts to submit some projects and protocols, but they not came to a fruition in terms of starting a study. I anticipate that, on the other hand, developing a vaccine is a challenge study. I mean, we do with our dengue vaccine that's under clinical development, and the chikungunya vaccine under clinical development. These are live viruses that we give to people. So it serves as a proxy to what we need to do in a challenge study here. Involves getting those pathogens produced in GMP conditions and submitted to all these pathways, and then having this approved for implemented the protocol. I think that's the overall view of the status in Brazil.
So, Esper, did you get the impression then that Anvisa would formally regulate the manufacturing process of a challenge agent and look at that? Or would they, so rather like the US approach, or would they just be supportive in helping you to work out a pathway? So is it actually a legal requirement in Brazilian law, which is I think where the difference is in the UK, because the operation of our regulator is only what has been written into law, and because it doesn't cover challenge agents, they don't do it because they're not funded to.
Yeah. So Anvisa doesn't have yet a specific guideline on human challenge studies. Nevertheless, the trend that we've been seeing for the past years is they usually follow WHO suggestions and requirements, and this is something that one would expect here once they are provoked. So usually we frame our responses and our pathways based on WHO requirements if they exist.
So I don't know whether Catherine wants to comment on WHO view on regulation, not the ethics part, because you're going to talk about that later, or are you going to cover regulation as well later?
No, I'm only going to cover the ethics.
Do you want to make a comment about the regulation? I don't know if someone could perhaps take a microphone for you, just about what the regulatory bit of the guidance says for WHO. So for David, who may not be able to see, this is Catherine Littler who's going to make a comment from WHO.
Except I'm not the regulatory, as David well knows, I'm not the regulatory expert, but have been working closely with the colleagues in the regulatory department in the development of our guidance. But the robustness, I think what's important is that regulation is in place. So all WHO guidance does say that in countries that the regulation does need to be in place, and that needs to be fit for purpose in that jurisdiction. And as we've heard out, there is huge variation in regulation across the globe. But I can't comment on the specificity because I know that's also being currently there's updates on the regulation pathways in relation to pathogens of pandemic potential, and also in relation to the look at the WHO list that's just been released in Rio, looking at the those pathogens on that WHO list. So I know there are discussions currently happening on WHO on the regulatory pathways in relation to those.
Great, thanks very much, Catherine. Unfortunately, I have to leave now to go to the airport, but fortunately Danielle has very kindly agreed to step in and to take over as chair. Perhaps I'll leave a parting question to Anna to give me time to escape, which is there were lots of difficulties getting Zika over the line initially, and I wonder whether you could go through that story of how we got from a point of a determination to do Zika, which was then blocked, and what the issues were that then allowed that to be unblocked. And then perhaps you can email me and tell me what you said.
Absolutely,
Thank
absolutely.
you very
much. Safe travels.
Safe travels. I will say from the start that it wasn't a regulatory hurdle that we had to go through. It was really around the ethics of a Zika chim. And I think that what I'm going to say next holds true for both regulatory and ethics, and that is the groups that are reviewing these protocols need to have expertise. They need to have expertise in challenge studies, and they need to have expertise in whatever purpose that challenge study is being used for. And the issues that we had in our review is that the review was put together very quickly due to some individuals who had concerns about a Zika human challenge, and said in public that they felt that it was like a Tuskegee study, which I don't know if you may not be familiar with that outside of the United States, but that's a very inflammatory statement to any principal investigator, I can tell you. And so the NIH convened a meeting. The ethical, the people who were on the ethics committee, although certainly experts in their own field, had never done a vaccine study, had never done a challenge study, were not infectious diseases people, and therefore, in my opinion, didn't understand why we do challenge studies, why we were proposing a challenge study, and what the role of a challenge study could be particularly for Zika. So the review happened in December of 2016, and the concerns that they said it's not time for a Zika human challenge. We can get everything we need to know from the outbreak, which at that time it was pretty clear that wasn't true because the outbreak was fading quickly and was almost over. And then just concerns around the challenge study itself showed they didn't really understand what we were doing. They were saying, well, even if you show vaccine efficacy in a challenge study, in your challenge study, you're going to have to do the same challenge study in pregnant women, which of course we would never, ever, ever do. And that was very frustrating. And, you know, essentially the conclusion was you need to wait and see. So everything was put on hold after that. And the epidemic waned. Cases became fewer and fewer throughout 2017 and 2018. And there was really nothing we could do at that point. We couldn't move forward because that recommendation was on the NIH website saying now's not the time. I have to say, you know, there was still great concern about Zika, and there was growing concern about how a vaccine would be evaluated for Zika, given that the only phase 2 study that was in progress was not getting any cases of Zika. So it was clear there was not going to be an efficacy endpoint in the DNA study that was ongoing in Zika endemic areas and in other places as well. And that's when FDA and WHO convened a second sort of consultation to discuss what is the pathway forward for Zika vaccines. And I have to say, I think for many different pathogens in terms and vaccines, I've found that bringing groups together and at several meetings over Zika that we had with Zika and other groups, we've had representatives from WHO and FDA at these international meetings. And David, you'll probably say this today because this is what every FDA person says. I'm not speaking for the FDA. But anyhow, it's been really, really helpful. It's been incredibly helpful to have these discussions. One, it raises issues on both sides. We can hear what the concerns are. They can hear what our concerns are, what we'd like to do. And it helps everybody, I think, move forward. And a lot of these meetings, as I said, are held internationally, and I think would be incredibly helpful if there were some way we can convene as we try to move a lot of these challenge studies into endemic areas, including Brazil, have these consultations where we have experts from WHO, we have experts from FDA, from EMA or even other regulatory groups, although if they don't regulate challenge studies, then it may not be all that helpful. But the groups that do regulate challenge studies, just to bring their background, their history, how they've approached these studies as they've evolved over time, I think would be incredibly helpful to the agencies that are learning about challenge studies, because I think as they build up their competency in reviewing vaccines and therapeutics and things like that, challenge studies are still kind of scary. To just hear about you're going to intentionally infect someone. But it was really that WHO FDA consultation that came out and said, in the pathway to licensure, a challenge study may be helpful. So it didn't say that it was it didn't say that it was mandatory. It didn't say that there would be approval based on a challenge study, but it essentially said challenge studies. The time is right. You know, we think that it's safe to proceed with challenge studies. We think there's a justification for challenge studies along that pathway, and that allowed us to move forward in the fall of 2019. And the review by the FDA of our initial challenge study protocol went quite smoothly, and I don't believe there was any clinical hold questions. I have to go back and look. I don't believe there were any clinical hold questions from the FDA on that protocol. Then COVID came and we had to wait some more. But, that's where I think really these discussions that you have with your regulators can be incredibly helpful as you're planning, as you're thinking. And if there's a way, much has been done with developing country regulatory groups. If even we could have a network regulators focused on challenge studies, or to learn about challenge studies, learn about how they're done in different countries. Bring in, as I said, people with experience from FDA, from WHO, from different regulatory authorities, from Thailand. So Thailand's done challenge studies. Malawi, Mali, they've done challenge studies. How did that work? Bring in those regulators to help talk with groups, country regulators who haven't done a challenge study, and really educate and then help them build their competency in that review, I think would be incredibly helpful.
Thank you so much, Anna. And I would ask you all to prepare some questions, because I'm going to open the room for questions to the panel as well. I think it would be really good to just discuss a little bit further on the Zika story with you and David. It's interesting, the word helpful in the regulatory pathway, right? And I wonder whether part of the discussions that happened at that point was the end point of your model, and whether in the way that you build your model was already constructed in a way that would be helpful for licensure. Was that part of the consideration in the discussion with FDA?
So that was part of the discussion, certainly in Zika vaccines. So the whole discussion around a Zika vaccine endpoint, what is your endpoint going to be? It can't be microcephaly, prevention of microcephaly, because the incidence is so low, your trial would have to be huge. So then the question was, well, is it just Zika? Well, how do you diagnose Zika? Because, and you saw from the challenge model, it's a mild disease. So most people don't have fever. So how are you going to identify cases of Zika in your follow-up? And one of the things that we wanted to do with the challenge study really is look and see, okay, what can we look at in terms of Zika shedding that may be helpful in, if you're going to do a field study of a Zika vaccine, if you had the incidence to do that. Because one of the things that were coming out during the epidemic was maybe you could look for Zika antigen in urine. We think that it's shed in urine for about 2 weeks. And what we found in our challenge study is that not everybody, the incidence by PCR even of Zika in the urine isn't high. Now that could be for a couple reasons. We use a very low dose. We were getting infectious virus titers of 3 to 4 logs in the Zika human challenge, so I would think that it would be fairly easily detectable in urine, but it's not. And I think that's, when you get all of that information and you think about what your challenge model is going to be used for, and ours was really developed, 1, as a possible pathway to licensure, but 2, to learn more about Zika. In terms of pathway to licensure, I think that it becomes more attractive because in the field, even if you had a large Zika outbreak, I think it's very difficult to detect cases of Zika unless you bring people back and think about it in your phase 3 clinical trials. Butantan's dengue vaccine study, they enrolled 15,000 people. You would have to see them like weekly and collect blood or urine to look for a diagnosis of Zika, because so often they're not clinically symptomatic. And that's where I think the challenge model can be very helpful, because when they got, when they were challenged with and you can follow them very closely to find evidence of infection based on viremia in the blood. And I think it would be very straightforward to design a trial for efficacy using the Zika human challenge model, and much more, I would say, feasible than any field study for efficacy.
Thank you. And I think it's important to mention that we have very few examples where, a regulator approval of a vaccine was made on the data from a human challenge study. And I can only say, the only example I can think of is Vaxchora, which I understand the FDA gave their favor approval based on a human challenge trial that was done for the cholera challenge model. Yesterday, we heard from Andy Pollard about the ongoing discussions for paratyphoid with WHO. But I want to really open the room, particularly for you all and David, about what do you think are the main obstacles we currently have to be able to push that a little bit forward?
I should mention, I think there's two ways that a Zika challenge model could be used in a pathway to licensure. One, I said, in terms of an efficacy analysis. I think the other is to identify a correlative protection. And if you've identified an antibody correlative protection, you can do a phase 3 clinical study to look at safety and also immunogenicity to see if your vaccine meets that correlative protection. I think with Zika, there's still a lot of unknowns that a challenge study will never be able to answer, and that is how much viremia is needed to cause microcephaly. And we're not going to go there with our challenge study, but I do think that that in terms of an accelerated licensure pathway, there are a couple ways you could use a Zika human challenge model. And David should comment.
Dave, would you like to comment, please?
Yeah, maybe just a couple of things just to reflect on the conversation up until this point. I think the one thing that I probably should have mentioned is we have a number of formal and informal mechanisms for engagement. Typically, folks take advantage of a type B pre-IND meeting, which is to ask the questions that you want to ask to avoid a clinical hold, right, and make sure everything's set up for that IND. And then after that, you can request what we call a type C meeting for all kinds of other issues that come up. Those are formal mechanisms. I will point out that there's no cost associated with those, and it's open to anybody. Even if your intent is not to run the study in the United States, you can still request a pre-IND meeting and get advice from us in terms of that study, both on the manufacturing side as well as the clinical and then the non-clinical studies as well. I think the other thing, and you brought it up, Anna, I think it's really important in the discussions with regulators to be crystal clear on the context of use. Is this in a translational research activity where you're trying to define a correlative protection or you're trying to do some sort of experimental proof of concept? That's a very different context of use in terms of a product-specific development, be it to make a regulatory decision, either pre-licensure, and in some cases you might think about it doing post-licensure to expand some indication or something of that sort. But being really crystal clear in terms of how that fits into a broader development program, particularly the clinical development program, is really critically important. Otherwise, I think there's a risk of talking past each other, where the sponsor has one idea and we have another idea and we're actually talking past each other. So what I would say is particularly in the pre-IND space, but all along, just be crystal clear. What is the context of use? How are you using that information, either to make product development decisions or you're asking for regulatory decisions? I think your responses will look very different depending on the context of use.
That's great. So I would like to open to the room now for some questions. Joshua has one. Yeah.
Hi, David. It's Josh Ozowicki. I think the last point that you made about human challenge trials as a clinical trials platform versus experimental medicine platform is really important. I think we're often falling into the chasm in between those two concepts. I'm not necessarily sure that we can resolve that always, and that there is going to be some tension and uncertainty in that space often. And I think Anna mentioned before, was talking about some of the unknowns that in fact are the reason for doing the study in the first place, and unfortunately also become the best reasons for not doing the study. And we end up just talking in circles around those two ideas. So, I sometimes think we just need to do the study and see where it lands. It's certainly, compared to the non-human primate studies that are routinely included in product development pathways, these are, we talk about the statistical fragility of efficacy signals coming out of human challenge trials with 30 people in each arm or something like that. You compare that to, non-human primate studies with 6 in each arm, typically, and see major companies are making huge decisions based on, 12 monkeys. And that seems to carry some regulatory, power. And yet these human challenge models we're struggling with so dramatically. And so I don't know that we necessarily need, I think part of the challenge that we have is that we struggle to define exactly what the product development, place of the study will be before we've done it. And I'm not sure that we always can. I think sometimes we just need to do the study.
David, would you like to comment on it?
No, not really. I mean, again, I would just suggest that you think really hard about exactly what is the primary objective of that study and be really crystal clear as you can be on it. Because I think that the guidance you're going to get back is highly dependent on that context of use.
Yeah, and let me comment too, Josh, because I think a lot of it is, and it may be dealing with companies. So if you do a phase 3 efficacy study, you're going to have a predefined efficacy endpoint that you want to see with set confidence intervals, right? And, you know, in a challenge study, I don't think you have to be that granular. You know, you can have where, you know, you don't have to, and I will say, because I primarily do phase 1, early phase 2 studies, and cover your ears, David, if you don't want to hear this, but, you know, I try to write my protocols pretty general. I try not to be so stringent that you have no flexibility in terms of, you know, certain things around your protocol, you know, certainly study days, things like that. But, you know, we tend to use a lot of may instead of will type language in the protocol, just because I think in early exploratory phases, you need more flexibility. You don't know what you're going to see. And again, you want to be able to get as much data as you can and use those data for for development, for future studies. So I think when you say, you know, see where it lands, I think you can write that into your protocol. Your protocol is going to be, this is why we're doing this study. We want to see if there's any benefit to this product. But we're not going to write criteria that are, if we don't see an efficacy of 60% with these confidence intervals, we're going to throw it out the window because, you know, again, smaller numbers, larger confidence intervals generally. So, you know, I think to David's point, it's really how you write those endpoints and what the purpose of the study is, and keep that in mind as you're developing your protocol.
Esper, would you like to comment?
Yeah, so I'd like to bring in one example for discussion. Mm-hmm. So we've been seeing that Brazil has been facing, together with Colombia, a challenge with the reemergence of Oropush. Mm-hmm. So this is a virus that we know, by and large, gives you a very mild disease, arbovirus, and the question is, when is the best time to start challenge studies with this virus? Mm-hmm. Because despite we understand that it's a mild disease, we had the same feeling with Zika before, prior to I think August or September 2015, when we thought it was just a benign, by and large, disease, but now we understand so much better. And when is the best time to start challenge studies for Oropush? And the second question is, how would regulators view this? I mean, would be acceptable today with the kind of knowledge that we have to start discussing? I think this is the answer to that would be yes, but to implement a challenge study for Oropush, not sure. What do you think, Anna?
No, maybe whilst you are thinking about it, I think it is a very good point, Esper, because I just want to bring the perspective of what happened during the pandemic in the discussions for the SARS-CoV-2 human challenge model development, where one of the lessons we learned is that obviously the discussion was extremely important and it happened, with a lot of stakeholders globally. But by the time we then decided to move ahead and produce the GMP virus, we were potentially a little bit late into the game. And one of the things that have been now very much at the forefront of thinking about human challenge for epidemics and pandemics preparedness is that if we were to have an investment pot where upfront we could already start the production of some of these GMP challenge agents, in parallel to the discussions about how would you do a study like that safely, and what the endpoints would be, that it potentially would be transformative for the human challenge field, because you would actually gain so much time and potentially have a model ready to down select vaccines or antivirals that could potentially be important in the containment of an outbreak and pandemic.
You know, I said this and in discussions with One Day Sooner and Jake early on, when we were discussing the SARS-CoV-2, I think people need to understand that you don't just pull a challenge model off a shelf. It has to be developed. And, you know, I think the first thing I would say to Esper, and I think the answer is yes, you know, do we have enough clinical understanding of Oropouche to develop a model safely? And I think that answer is yes. You've sort of described what the clinical illness is, and I would say absolutely now is the time, as early as possible, because you do want to have a model ready that can help vaccine developers or therapeutic developers. And, you know, when I'm reading in the press some of, you know, the congenital anomalies that they have been seen with Oropouche, I think there is going to be a demand for a vaccine or interest in the development of a vaccine. And I think now is the time, and you can have that model on your shelf. It can be, you know, available. As long as you've fully characterized it, that will help developers understand how to design their trials depending on what their endpoints are going to be. And so I would say yes, if you can generate interest in funding to have a program for development for that, I think it would be very, very useful for future products.
You would start with the translational study,
Yep.
and then set the framework for intervention studies.
Yep.
Khalil?
Yes, well, some of you here were part of this meeting that we had in Geneva to prioritize pathogens that could be dangerous. And I think, David, you were there. Anyway, in CEPI, last week, this was publicized, because so far they wanted to work a little bit more before publicizing the pathogens that could be dangerous. So from that list, many of them, it will be very difficult to have a vaccine and test it if you don't have a challenge experiment. So how we're going to do this? I mean, we have been working this RB blueprint for WHO for many years, and we have reached a prioritization of pathogens. What's the step forward? Should we choose some of them and try to do vaccines and do challenge experiments? What would be the next step?
Would you like to go first? Yeah.
I can try. You know, I think one, you know, every pathogen is not suitable for a challenge model. I think we have to start there. So looking at the list of pathogens, you would see, I would say, for a traditional challenge model. But, you know, can you make, for lack of a better term, pseudo-challenge models? So can you make recombinant viruses that express what we think are the protective antigens of the pathogens for which you can't really have a true wild-type challenge model? So, you know, I think, and what's interesting, I think, as everything evolves, I think the regulatory process around this, around accelerated pathways, are going to evolve as well. If you look at, you know, for these pathogens where we don't think we can do traditional phase 3 efficacy studies. And, you know, chikungunya is a good step forward for that, using an immunocorrelate, an antibody correlative protection. You know, you still have to do those follow-up phase 4 studies to validate or to show that your presumed correlate was in fact relative. But I do think, you know, looking at that priority list, you could then look at, you know, sort of in parallel, working with regulators, okay, what would a regulatory pathway for this particular pathogen look at? What are the steps that we can do? What data, what reagents, what program do we need to get ready so that we have candidates available? And I think looking at human challenge studies, there are some of those that you could, as I said, either a wild type pathogen or you use a pseudo wild type pathogen to try to develop either the efficacy model or immune correlates are something that you could use for that regulatory pathway. And as with everything, the earlier you can start, the better, because you don't want to be reactionary, you want to be proactive. You want to have something ready if you have a product. And I know that CEPI is working very hard on their list to get vaccine candidates moved along for some of these priority pathogens. And I don't know, it's interesting, David, maybe you can comment on this. I don't know if CEPI has engaged WHO and other regulatory authorities on, okay, we're going to have these candidates ready. What's the regulatory pathway for them? Is it just we're going to test them in an outbreak setting, in experimental design? Are we going to do our phase 3 studies sort of in the image of Ebola, in the ring vaccination study? But I think it's important to have some sort of guidance or available so that when the time comes, there's people can move much more quickly rather than trying to develop these studies on the fly in the middle of an epidemic. If you can have something in place to say, this is what the regulatory pathway we think is going to look like, this is what you should be prepared to have ready, this is what you should have, sort of different elements in your clinical program design, I think would be very helpful.
Well, if I can comment on this?
I don't know if David has a comment before that.
Yeah, just really quickly, I think there's been a number of different concepts that have been thrown out, and I think we need to be really quite clear. To answer your question directly, Anna, absolutely. We're having discussions in terms of the regulatory pathways. What I would say at a high level that's come out of that is, and Andy started off by saying this, there are different regulatory frameworks in different jurisdictions. And I'll just maybe take a couple of seconds here to talk about one that you have mentioned a couple times, Anna, which is accelerated approval. That is a specific expedited program within the United States. I don't know actually that exact mechanism actually exists in any other jurisdictions, but just to tell you a little bit about that, because I think there's elements of it that are critically important for this discussion. There are 3 qualifying criteria to meet to go down the accelerated approval pathway in the United States. First, it needs to be a serious condition. Secondly, it needs to be a meaningful advantage over available therapy. And the third is you need a surrogate reasonably likely to predict clinical benefit. And you still need, the statutory requirement is still a substantial evidence. So the statutory requirement for evidence doesn't change. It's just what type of evidence you're bringing forward. But the condition for that, which is I think critically important, is you have to come forward with a feasible post-confirmatory studies that can be done with due diligence. And in the case where it's hard to run a randomized controlled clinical trial pre-licensure is going to be a problem for you in terms of designing that post-confirmatory study, because if it's not feasible to do it pre-licensure, how are you going to do it post-licensure? One of, I think, the regulatory frontiers for us is how do we use real-world evidence to fulfill the post-confirmatory study requirement? I think we're in new ground here, but I would think that needs a lot of thought. And I think that thought needs to occur pre-licensure rather than in the approval process, because otherwise it'll really slow you down. So I just maybe highlighting that point. Over.
Yeah, the quick comment I was about to make is taking the chikungunya example. I mean, the 2 studies required by the FDA, one is a pragmatic placebo-controlled trial, is in a ballpark of 20 to 30,000 volunteers. In the phase 4 is 500,000. So the amount of resources that we need to allocate is really robust. I mean, in these post-marketing studies.
You had a comment, George?
Yes. I think that this tremendous effort to try to identify what would be the dangerous pathogens is just a start. And from the several families that have been studied, that were identified some families that could be more dangerous, and we have selected the one that would be the prototype of that family. And I think that next step we should see from those which ones we could go further. I mean, how we can study the immunology response, how we can have protective correlates of protection, and for some of them, perhaps we should try to do a vaccine and keep it in the fridge. There is some regionalization of that. That means that Oropushi, for instance, is important for us here. It's not important for North America, for instance, and Europe, but there are others that are important there. But I think we should prioritize, and this should be a guideline for agencies that are going to support for development of vaccines, CEP included, but then others. But I think it's a tremendous work that has been led by WHO that we should take in consideration.
Agree. Elton?
Hi, David. This is Helton Santiago. Thank you so much for joining the table. It's been a very rich discussion. I would like to go back to the Zika vaccine specifically. FDA has approved a vaccine before just based on CHIM efficacy. We have talked a lot about the surrogates markers of protection and what it would take to that CHIM can be used as evidences for regulatory approval. But regarding to the Zika and knowing that a traditional phase 3 will be impractical, I would like to know if you, as a director of FDA, I know you're not speaking by FDA, but if you would have any thoughts about the best approach to license a Zika vaccine with the situation that we have in the epidemiological situation, and the necessity to use CHIM to show some evidences that will allow the use of the vaccine.
No, thank you for the question. It's a great question, but one very difficult to answer. What I would say is the approach typically is to look at the totality of evidence, that we just don't focus on one piece of evidence. We look at the bigger picture. So in the example of where it's been licensed with a CHIM study, that wasn't the only piece of evidence that was brought forward to support that licensure. There's a whole body of evidence, mechanism of action, experience with similar vaccines, et cetera, that paint a bigger picture that allow one to make a regulatory decision. I think in order to opine on the role of CHIM in Zika, it is going to be critically important to see what is the totality of evidence that's going to be brought forward to say that you have addressed the uncertainty to a point that's acceptable to approve it. We're not going to reduce all the uncertainty. There will be some residual uncertainty, but you need to, through the totality of evidence, have reduced the residual uncertainty so it's acceptable and there's a favorable benefit risk assessment. And I hate to say it, but I think it really is true. It's on a kind of a case-by-case basis in terms of what is the evidence for that specific product, right, that says that it has a favorable benefit risk assessment.
And I would go back, David, to what you said earlier. I think that's for really early engagement with the FDA in terms of coming up with how you're going to design your study to get that approval using a CHIM model and really start discussions early on to figure out what might be the most expedient pathway or the most successful pathway, I should say.
Okay, thank you so much.
Maybe just to put a finer point on it, the statutory requirements here in the United States for traditional approval has typically been two adequate and well-controlled studies. In the vaccine area, what we've been able to do is to say you can get by with one adequate, well-controlled study, usually multi-site, usually very, very large, with other confirmatory evidence that support the outcome of that one adequate, well-controlled study. So I think you just need to keep that in mind as well. That's the statutory requirement. That's the substantial evidence that's needed for traditional approval.
Yeah, and I think the other point to make, and it may come into that too study, is even if you do a CHIM and you show efficacy, you have to do a sufficiently sized safety study in addition of your product to ensure that it's safe as well.
There was a question there, Jake.
Hi, yeah. So I have a question. So Anna, you've talked about kind of two ways the model can be used for licensure. One is this designing probably larger CHIMs, Zika CHIMs for efficacy testing, and the other would be identification of correlates of protection that you can then validate in the field. Just from like an operational and mechanistic standpoint, like are those two different sort of, like those are the same model, right? Can you do kind of two birds with one stone where you're testing vaccine efficacy and then finding those correlates of protection and then using, the results from those two, from, that series of studies as kind of, I guess, both sort of, separate parts of the totality of evidence that an organization like FDA is looking for? Or do you think that would be slightly different sort of ways you design these studies and probably different funding requirements and stuff?
So I think if I had all the money in the world, no, I think you certainly, you can have multiple outcomes to your study. So to design a study for efficacy, you would have what is the dose of vaccine that I have chosen? What is the dosing regimen that I have chosen? And what is the outcome in terms of efficacy outcome that I want to see? And if you were just to do it for efficacy, that's what you would do. But you could do a large study where you say, this is my dose that I'm choosing, this is my dosing interval that I'm choosing, and this is my outcome for efficacy. I also want to see if I can find a correlate of protection. Now, in some studies, you could do that with just your efficacy. You would have some breakthrough infections. In theory, you would have some breakthrough infections, and you could then do an analysis if you've powered your study well enough to say, okay, we saw breakthrough infections for antibody titers less than 1 to 100. Everybody who had an antibody titer greater than 1 to 100 was protective. We have our analysis. You can also do something far more granular where you actually push your vaccine to fail so that you can have, I would say, a more granular assessment of what is needed for protection. So you can include in your study multiple doses of your vaccine going lower, 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 pushing that vaccine to failure and seeing if you can find something more granular. So you could do both in one study. You may be able to do it, as I said, just looking at one dose, one dosing regimen if you have breakthrough infections at that dosing regimen. I would prefer to really do it more stringently, just from a scientific point of view, to see if we can get a real granular. Now, I will also say, though, and we say this a lot with different vaccines, the correlate of protection you get with one vaccine may not be the same for other vaccine platforms or other vaccines, and that has to be kept in mind as well. It's when you do that study for your vaccine, in my opinion, that's for that vaccine. I mean, we know this for dengue. Dengue's taught us this very, very well. Even if you have live attenuated vaccines, the correlate of protection for each serotype is going to be different, and for each vaccine for each serotype will likely be different. So it really is, when you're doing it, it can be very narrowly applied, in my opinion.
Any more questions or comments in the room?
Yeah, just to pick up on that, because I think that's a really important point. And I would also say it also depends on the indication that you're seeking. Depending on the clinical endpoint, that correlate of protection may be different.
Agree, very good point. I just want to go back to one point that Esper made earlier that I thought it was a very interesting thought, make it a bit more general, which is we probably have been doing a lot of human challenge studies with vaccines in Brazil for a while, right? We just don't do the readout. Yesterday Ben showed the Valneva vaccine with such a beautiful viremia curve, very safe vaccine in the participants. And I think that that's an important thought, particularly in an environment where that's one of the reasons for this workshop about the debate whether, could we or should we do challenge studies in Brazil. I think the idea that with very safe vaccines, we haven't been doing that with live attenuated vaccines for a while is an important thought to just hold for this panel. I don't know if you want to comment further, Esper.
No, absolutely. I think there are several uses for, challenge studies and in taking the vaccine side of the story. One example that we had recently discussion is developing monoclonal antibodies to prevent infectious diseases, especially, tropical and subtropical infectious diseases that are more important to low and middle income countries. And the example that popped up was yellow fever, for example. We do have some candidates coming up, and what would be the best challenge study? And we do have our yellow fever vaccine is a challenge that we do every day, to people. So the only way out that we found to trying to answer that question is, would a monoclonal antibody prevent seroconversion induced by a vaccine after being given, after being with pre-treatment of any candidate monoclonal antibody? So there are several examples that we can utilize vaccines and use this platform as a surrogate, not a surrogate, but a model to try to answer important questions.
Yeah. I'll follow up on that, and Catherine may also want to comment because I think she knows a little bit about this. So Wellcome funded development of challenge models in endemic countries, and Oxford Group was developing or trying to develop a human challenge study in Vietnam. But there was a law in the books that said you could not deliberately give an infectious agent to a person. That was a criminal offense. Well, we give live attenuated measles, mumps, rubella, and that was sort of a pathway into discussions with regulators and with lawyers and government officials in Vietnam to say, look, we do this with live attenuated vaccines. The challenge studies, they're well described, and it was dengue in particular, but that use of referencing vaccines was sort of foot in the door to discuss that with legal authorities and legal scholars in Vietnam to try to make a pathway toward challenge studies in Vietnam.
Thanks for sharing that. That's really, really helpful. Any more questions, comments?
I haven't. The pandemic came, and so that was. She was making progress. Bridget Wills was making progress, so it did have some effect, and they were making progress. I don't know if the grant has ended though, and I don't know. The grant ended. Yeah, I think the grant ended. So we don't have a challenge model in Vietnam yet, but I think progress was made.
I have another question. Hi, David Demert. This is kind of totally unrelated to what you're just talking about, but just something that came up at the beginning, at least in the US, how it's a requirement that the challenge agent is made according to GMP. I wonder if you could, maybe the panel could just comment on that a little bit more, because I think when people hear that, they get really scared off and they think, oh my gosh, it's going to be really expensive. I have to hire some company it's going to cost a lot of money to make my agent. But as you said before, Anna, that there's guidance, and that we've already developed 2 challenge agents just at George Washington University, following the guidance on our own. It, of course, did require a lot of time and effort and documentation, but GMP does not always mean super expensive.
I think you should comment on and I know you mentioned it in your talk, but just to review sort of how you did that, because I thought that was really, very, you did an excellent job. And you're right, people get scared off when they hear GMP. They think it's very expensive. You have to send it to a company to do all of this. And just how did you use the guidance to come up with your strategy for completing the GMP requirements?
Well, first I'd say that it wasn't me who did it. So Jeff Bethany at GW was responsible for it. We did have a consultant that we hired, but again, that doesn't cost a ton of money. And it was mostly a lot of documentation. I think it does obviously depend on what your agent is. So, we're, producing larvae or eggs for intestinal helminths. It might be something completely different for a viral agent.
And how it's administered, because it's
sure.
different for giving it intravenous or versus intranasal or skin. Yeah.
Definitely, yeah. But a lot of it was, putting all of the documentation and controls in place and, developing our release assays, most of which we could do in-house. We did have one, the bioburden testing that we had to send out to a commercial lab, but sending a lot or batch out for just one test doesn't necessarily cost a ton of money.
Can I just comment on that? So, because that's exactly what we did for the pneumococcal challenge agent as well. And for 15 years, we have been producing very effectively and safely several batches of the bacteria by using the same process, which is very much about the documentation. And we work with UKHSA in the UK to test every single one of the batches and certify purity and sensitivity to the antibiotics for us as an external laboratory. But the whole process about breaking down and saying, every single one of the steps, how do we ensure purity and document everything. And at the end, it's not extremely expensive, but it's extremely important for our program that we were able to do in that way, because then we could test 15 different strains, which if we were to pay $1 million each for a company to produce for us, would have completely hinder our program.
Yeah.
So I wonder if we don't have any last comments or questions, we could then break for coffee. I would like to really thank our panelists. Thank you so much, Dave, and thank you all of you for joining us. It was a very rich discussion. Really enjoy it. We went beyond Zika and went back to Zika, and I really, really like it. So thank you so much, and we're going to have a coffee break now for half an hour. Thank you so much, everyone.
Thank you, David.
Bye-bye. Have a good day.
roundtable of ethics now. And we will start with Catherine Littler, who is the co-lead of the Global Health Ethics and Governance Unit at WHO headquarters. Her work focuses on intersection between ethics, governance, and policy making in global health research and practice. I just learned that she has a law background, which is very suitable for ethics. The bioethicists that are helping us in the project and are going to speak also this morning from Brazil, they are all from the law school. So, and the importance of Catherine here is that she led the guideline of WHO on ethics for CHIM studies in endemic areas. So this was a very important document that is helping several countries to establish CHIM models with safety and with the security of doing the right thing and not messing up. So, Catherine, thank you so much for your work. Thank you so much for taking your time and being here. And the floor is yours.
Thank you very much. It's an absolute pleasure to be here. I try to keep it hidden that I'm a lawyer. I like to say I'm a reformed lawyer. And also the caveat should be that just because something's legal doesn't mean it's necessarily ethical. I think it's also important to say. So thank you, Daniela and Hilton and Anna, for all your organization. And also Lucas, who gave the talk yesterday. Oh, there he is. Thank you for also giving the historical context to challenge studies, because I think it's important, but I'm not going to go back over it. I think the key point about the historical point, and I was very sorry to hear what was said to Anna earlier about a reference to the unethical trials of the past, is I do think challenge trials now currently are very different to the unethical challenges that were taking place in the past. So I think that's a very important opening caveat. Apologies in the room for people who are very familiar with all of this, or the people working on the field. I'm going to try to give a whistle-stop tour of the work that we've been doing at WHO since roughly 2019 to develop human challenge studies guidance. And you'll see we've got 3 different areas in which we're working. So the first one, the perennial, what we're calling the perennial pathogens. This is the guidance that we've worked on that was published in January 2022. And that's the guidance focusing on endemic settings, and that's looking at pathogens that we've been talking about, so covering dengue, Shigella, typhoid, TB. Interestingly, we don't have a hookworm case study, but maybe we will need one going forward. So that's the one that's already published. The second one is guidance looking at doing challenge studies during emergencies. So I'll talk about the guidance we produced during COVID. But we're also currently working on challenge studies, generic guidance for doing challenge studies in emergencies. So that's pathogen agnostic. That's in the final stages. It's gone out for public consultation, and we are finalizing that, and that should be published depending on the WHO review process. For all those familiar, I'm hoping September, October. And then finally, something that's come up in the discussion and the panel today, there is a lot of interest in doing challenge studies for very good reasons on what we're calling pathogens of pandemic potential in the interpandemic period. So this is really focusing on those families of pathogens or viruses that are on the WHO pathogen priority list, but also looking at organisations like CEPI and their list, because they are really moving into this space. And there's a real sense of urgency here with these 100-day missions. So I think a lot of funding is going into
looking at these types of challenge studies. And we're really interested in developing guidance in that space. And I want to pick up on the point that Anna made. We're going to really extend that guidance working group. We know that in order to do good ethics guidance, we need scientific expertise. There's quite a large family of priority pathogens. So it's going to be tricky for us as to how to bring in all that expertise. So I'm going to focus on these 3 today. Well, there's 4 actually, the one we've completed and then the ones we're working on. I'm also going to make a couple of reflections based on the experience of doing the guidance and trying to actually analyze where the uptake is, how effective, and I really want to focus on learnings from that, particularly for your context, doing these types of studies in Brazil. And I'm really thankful that I'm being followed by Primus, because I think what's going on in Kenya is an excellent exemplar of what you could look at to learn from Brazil. A lot of work, a lot of detailed work, and maybe one of the lessons you may not like is some of this stakeholder engagement and socialization. It will take you time just to familiarize people in the first instance, but I think once you've got over those hurdles with both populations, so it's not just, engagement is not just about populations, it's about also ethics committees, regulators, and interestingly enough, politicians. Because they're the ones who might make knee-jerk regulation if they don't understand the science here. So moving on, I'm going to talk about the first guidance and why we actually put this guidance in place. You've heard a lot about WHO and the SAGE recommendations and the fact we've got regulatory guidance on challenge studies. So a lot coming out of our technical teams. But at the time of putting this guidance together, we didn't have any ethics guidance. We were getting calls from member states asking us, because there's growing interest in doing challenge studies in endemic settings. They were asking us if we could produce some guidance. We were also getting calls from the research community. So that's the backdrop. We know that challenge studies have been going on a long time, as was pointed out yesterday. I think statistically is that it's over 70% are in the US and the UK. So a long time in high income settings. So the focus of this guidance was really on doing challenge studies in endemic settings, which are often low and middle income country settings. Not always, but often. And as climate change has an effect, that may not be the way that it is in the future. So really a focus on low and middle income country guidance. It's really important to say that this is complementary guidance to pre-existing ethical guidance that exists out there. This should sit on top of the SIAMS guidance that's out there, and also the Declaration of Helsinki, which is actually being currently updated by the World Medical Association as we speak. That will have a section on participation research with healthy volunteers, obviously of which challenge studies are in that category. So we have been engaging with them to make sure what they update is complementary to the WHO guidance. And you're going to have to stop me at some time, Helton, because I haven't timed my talk. So I'm just warning you. I'm on Brazilian time, not Swiss time. What it is, it contains case studies. I'll show what they are in a minute. And some people in the room were involved in the development of those case studies. And actually going forward, what will be really interesting is to work those case studies into questions for regulators and research ethics committees. So I've actually learned quite a lot on what we could do better. So we'll update this. There is checklists for RECs. But again, we know they're being used by some research ethics committees and IRBs, but actually I think we have to be cleverer with the way in which we put those checklists. Interactive tools would be much better, I think, for uptake. There's additional information on consent. I think one of the key messages I want you to take home is that consent is fundamentally important in here, and I think Noni and Primus will talk about the fact that there are tests for comprehension of consent going on in challenge studies that I think are critically important. Consent is not the only thing we need to rely on, but it's important that it is incredibly robust in challenge studies. And then the COVID document is also in an annex of that. So moving on, the key premise, I've already mentioned this, we are arguing that challenge studies aren't fundamentally ethically different from other types of clinical trials with healthy volunteers. They are a continuum of the spectrum. So that is the starting premise of this guidance. So we're not exceptionalizing them in other ways. That's important to understand it. And that, again, the guidance is consistent with these themes. The guidance addresses when and how CHIMs may, and I'm saying may be conducted. I liked when Anna said the use of the word may, not when we may do this, not going to do it. So we look at the issues relevant to clinical trials, issues that are morally significant and require particular attention in challenge studies. So risk-benefit would be one of those issues that we would focus on, and scientific justification is another one. We look at the types of infection models and the risks and burdens. Where there's area of consensus, I'm going to come back to this, because there's lots of areas in the ethics community in challenge studies where there is not consensus. Okay, that's important to acknowledge. Where there's debate, there's lots of debate still going on where these are acceptable, and also where there are levels of uncertainty. And what is acceptable uncertainty, I think, is a really important question in challenge studies. It's an important question in science. And finally, we have a list where we think further research is needed. As this is an ethics document, it starts with the core values underpinning this. And I think the one I really want to draw, obviously the procedural runs around transparency, honesty, accountability, and inclusivity point to the governance. Social justice and global health justice really point to issues around access and benefit sharing. And I hope these will come up in the conversations that happen after mine. But I really want to talk about, or just draw your attention to something that I think is really important, and this is responsible stewardship of science and of research resources. We cannot just put the burden for the ethics of doing challenge studies on research ethics committees. They must be integrated into the design of the research at the earliest possible stages. So we would advocate for ethics advice in the trial design team, as Helton has pointed out is happening in Brazil at the earliest possible stages. It's too late if you haven't sorted out your ethics questions by the time you get to a research ethics committee. So we have to rethink the way that we do ethics in order to turn it from something that's considered a policing mechanism to something that's considered an enabling mechanism of quality ethical research. These are the sections of the guidance document. I don't have time to go into each of them. As I said, with the risks, burdens, and benefits, that's really important. So what are the kind of benefits you would focus on? You would focus on the scientific justification. You would focus on the generalizability of that information and the public health benefit. The risks, minimizing harm, would be a key risk that you would focus on. And also looking at the degree of uncertainty in the research and whether it's an acceptable degree of uncertainty. Just to show you a bit on justification as one area, as I said, I don't have time to go into each of these individually, but scientific justification is a critically important one. Questions that you should be thinking and reflecting on: is this the only way to address this, or is this one of the major ways to address this? Is this the most robust and rigorous? Is it the most expedient? Is it the most cost effective? Will it involve fewer participants? I mean, that seems like a no-brainer, but it is a fundamental question. Is it lower risk than alternatives? And if it's not, how do you balance it against the ones above this? And is it a critical component of the research pipeline? So when Ben was speaking yesterday, it reminded me about, I was in one of the TB discussions that he was talking about. It reminded me about one of the discussions that was happening on the TB with the regulators. If that data is not included in the regulatory pathline, then some of these TB challenge studies may not be ethical. So it's a fundamental about where that data, and discussions with regulators and ethics committees are important to iron out those types of issues. As I said, we've done case studies in these. These are the ones that we worked on, but I've even heard of today and yesterday some more that we need to focus on. We'll be revising these. We'll also be doing with the additional guidance that I talked about, the guidance that I'm going to come up with a much catchier title, but the ones on the pandemic of pathogens of pandemic in the interpandemic period. We'll be doing a whole series of case studies. But actually what I've learned from being at a few of these meetings is that we need to building questions for regulators, research ethics committees, and researchers into these guidance documents. Importantly, we have a section on consultation, engagement, and social science, and I know Noni's going to pick up on this. And Primus was actually involved in the scoping review that we thought was critically important. So there's a scoping review of engagement, and there was also a scoping review done of social science studies that's in the back of these documents. And this was really focusing on what's happening in low, middle-income country settings and endemic settings. All of this is in the guidance at the back. As I said, when we developed this guidance, we realized there are a lot of unsolved questions that we couldn't resolve in that guidance development working group. So we've put them out into the public domain as further research. I think some of them are being picked up by some of the research groups that are working on this. So questions around acceptability of research with, I would say, vulnerable and potentially marginalized groups. And I put vulnerable in this sort of inverted commas, including pediatric populations. And this is a big debate, where it is acceptable to do it in pediatric populations, and what are the perceptions of trade-offs in the research design amongst stakeholders, issues around risk, benefit, and harm that need further exploration, including how are risks to third parties perceived and experienced by stakeholders. And this is based on both the 2 years of consultation on this guidance and those scoping review. This is where these questions have come from, as well as the experts we consulted. Perennial issues around consent. Consent, I think ethicists could spend their lives on consent. But these are fundamentally important questions, especially around how risk should be discussed and considered. And hopefully this will be picked up by the following speakers. And then other issues around withdrawal study and incentives. Reimbursement and incentives is one, I think, in which a lot of work needs to be done, because I think it is very contextual. What is acceptable and what's undue influence in different settings will look different. And then obviously review of research, which has come up quite a bit in today's discussion, both in relation to the hookworm example and also in relation to Zika. Moving on to the guidance that we developed during COVID-19, really looking at challenge studies. For those of you who know WHO guidance development, this is actually quite fast, not in a pandemic though, right? So I really want to make a call out that when we're talking about pandemic preparedness, that doesn't just flow to the science, that flows to the governance, right? We need to be better prepared. In an ideal world, we would have had this guidance, pathogen agnostic, already in place, which is why we're developing the pathogen agnostic guidance already. But actually, the timeline is quite fast. The challenge studies were mooted in 2020, but they didn't actually take place until 2021, and we were able to move quite quickly because we had this pre-existing working group already in place looking at the generic guidance. So that's where you'll see the timeline fits in. So these guidance were ready and available for the UK government's, the UK Health Research Authority's Ethics Committee to consult this guidance. We've heard a little bit about the context of where we are. So where we are now is really thinking about the next public health emergency and what we need to have in place in terms of ethics guidance. The context of this guidance and how it differs to the generic one, clearly higher levels of uncertainty, higher community and transmission, but at the same time, larger potential public health benefits of doing the challenge studies, and increased research funding and activity. The amount of money that went into COVID is phenomenal, and the amount of money that could potentially go into challenge studies. So really, we focused on the coordination issues. We did not want to see any kind of duplication of these types of studies. But along with that, potential shortages, which is exactly what we saw in COVID, of healthcare resources, and a lot higher level of public interest and scrutiny in science more generally, but also in challenge studies. This has, I think, positives and could potentially have negatives if you're not prepared. So this is, in essence, the key criteria document. It's quite similar to the other document, but I would draw your attention to the coordination point. We felt very strongly that there had to be coordination, including with a government of the country in which a challenge study was approved, because the uptake into the response was fundamental to it being ethical to do it. So we really thought this coordination point. We also thought that the sharing of the challenge strains was really important. The sharing of any preliminary data had to be done fast because of the public health imperative here, and we didn't want to see any types of duplication. And then the other criteria that's different to past is, you'll see number 7, we did think that there should be expert review, independent expert review done on challenge studies. So they're the two fundamental differences really in this document than into the generic document. I'm not going to go through, I've already just mentioned the coordination. We then followed up with the UK Health Research Authority. So the UK is the only country that authorised a COVID challenge model. And we worked very closely with the UK Health Research Authority after they had approved this to discuss the benefits of our guidance and how they used it. And the conclusions were that the guidance, there were elements that they thought was too generic, so we're trying to address that in the next version, but they considered the guidance useful in a real-world setting for both the researchers. We met in that follow-up meeting with the researchers whose research was approved, the Health Research Authority, and members of the UK Ethics Committee. It did at the time represent an international consensus document. We gave the ethical criteria. We didn't say you can do challenge studies. We just gave the criteria for doing it. And it did set a powerful role for norms and standards. I think having this type of guidance is critically important to have in any type of outbreak, which is why we're following up with the 2 other guidance that I mentioned at the beginning. Some learnings from COVID in terms of challenge studies particularly. Consensus on the use of challenge studies is incredibly hard. If you did a little poll with ethicists, I think the majority of ethicists in the UK would say that this is fundamentally ethical, the challenge study that was approved in COVID. A lot of ethicists, not all, in the US would not say that they would find this. So it's hard to get consensus on that. Context matters. I would argue that maybe the UK is probably one of the very few countries in the world in which you could do a challenge study, because it has the NHS, the National Health Service, right? So follow-up, in case there is long COVID, can be conducted. So it may be unethical to totally do this in a country that doesn't have a National Health Service because of that. So context of where you do that really matters. The model of what they did in the UK, I think, was excellent and needs a form of replication. And I've added the word form deliberately because I don't think you can emulate that kind of engagement in every setting. Different kinds of democratic structures, different kinds of resource, different kinds of availability. But it'd be really interesting to ask people who are doing these models in countries like Kenya, what kind of engagement would you do there? But I think the levels of public engagement is critically important, as well as the investment by the UK Health Research Authority and the Research Ethics Committee. So they trained that Research Ethics Committee. They had at least two separate days where they brought the Research Ethics Committee together, explained what a challenge study was, gave them the opportunity to and fro to ask questions. I think that level of understanding before actually having the protocol in front of you is really important for both for complex trial design. So I would argue you need to do this for things like adaptive platform trials where the Research Ethics Committee is naive to the process or the style, but also for these types of things. And I think maybe that's a valuable lesson for Brazil in terms of going forward and discussing with CONEP, having these types of pre-discussions where they can really air some of the understandings of what they don't understand about the trial design or what are the issues people have with them. And I think we'll hear from Jake later, but I don't think you can underestimate the growth and amplification of voices of people who wanted to participate, so the One Day Sooner movement. And it will be interesting to see what kind of impact that has going forward. I think I am running out of time, so quickly then in terms of the ethical lessons that we've learned, I think the others are ethical, but just really, I've said it before, I think it really is important to emphasize that modern CHIMs are very different to the historical CHIMs. They are very robust, very well governed, very well regulated. That CHIMs or CHIS are sometimes justifiable clearly in the vaccine development process, as we know that the challenge study for COVID was actually approved after the vaccine development. So it can be, the vaccine was developed. The scientific justification needs to be strong relative to alternative designs. Consent should be high quality. Payment for time, in the UK at least, is acceptable. Be interesting to have these discussions. I know that in Brazil you even have regulations about compensation, so you're limited in scope by that. Risk to volunteers and third parties clearly should be minimized, and emergency challenge studies can be ethically justified. So maybe if this was done, the Zika came after the COVID, it would be a different story, I think, now. And then some unresolved issues. At the beginning I said there isn't necessarily consensus. I think we do need to do more empirical research. We do need to do more engagement. These are some of the things in which I would argue we knew. So can non-vaccines benefits justify challenge studies? The question on upper limit of risk is a really tricky one, and I think depending on who you are, people feel very differently about that, and I think we need to have some open discussions between scientists and ethicists about upper limits of risks. Lasting harms, long COVID, is that an acceptable side effect of doing a challenge study? How much third-party risk is acceptable? How should uncertainty be evaluated? This is really tricky. I don't think this is unique to challenge studies, by the way. The question of uncertainty in COVID really came to the fore, and I think the public for the first time really understood the role of uncertainty in science. And then what are optimal approaches to community engagement, regulation and review? And I think we'll be hearing more about optimal approaches to engagement from Noni and colleagues in the next session. I also think these kind of discussions are fantastic, but you have to also situate CHIMs in the broader picture, because there's a lot going on out there. I think the clinical trials resolution is really important, because in some ways this will help advocate for challenge studies as we look to reduce research wastage. Huge amounts, thousands of trials underpowered during COVID. Millions of dollars wasted. I think challenge studies will be promoted as an avenue that can help with that. Investment, I really want to make this point incredibly strongly. Investment in ethics oversight is key. Often research ethics and IRBs become the punching bag. You are the delay, for all sorts of reasons. But we brought 30 research ethics committees together from across the world at the end of the pandemic. Almost none of those national research ethics committees had any additional money put into them during COVID, right? So all that additional money that went into research, none of those research ethics committees got any money, yet the amount of research applications increased dramatically. They often had no paid staff, and all the people doing the reviews were researchers, right? So what do the researchers do first? They don't do the reviews, they do the research, right? So I think we really have to be prepared. When we talk about preparedness and response, we have to really think about how we invest in research ethics governance. And as part of that, we have a whole program of work looking at benchmarking research ethics committees. How do you tell if a research ethics committee is a good research ethics committee? You have to have some benchmarking tools. Also looking at mutual recognition models. How can we, for example, on a challenge study, maybe in an outbreak, like they did in the UK, you just have one committee designated to look at that, and that committee is trained to look at that. So at WHO, we had a headquarters ethics review committee. At the time I had responsibility for. I set up a committee of that just for COVID applications, and that committee was full of infectious disease specialists and ethicists who'd worked on infectious disease, and that was deliberate because they didn't have to get up to speed. So we could turn applications around in 48 to 72 hours because they had the prerequisite knowledge. So I think we have to be clever, not just about the science, but about the oversight of the science. So we have to be adaptive and flexible. And I really think we should think about specialist committees, and I think that's a job that we at WHO are doing. Regulation is reassuring for research ethics committees. Let me tell you that they like to know that there's a legal boundary there, and that's really important in any type of research. I don't think we can underestimate the impact of vaccine hesitancy and the fear of doing things like challenge studies. So I think it's really important to have advocacy tools and to have programs of how we engage on challenge models. This is the one we're working on. I'm not going to go into this because we're running out of time. Final reflections, because I think there's a lot of good South-South learning that needs to take place. Issues that are considered particularly salient in LMAC settings. Consent and education level, I think we've heard a lot of the time that the participants in challenge studies in high-income countries are obviously poor medical students, but that's not the case in endemic settings, so questions around that come up. And what sounds like it has come up in some of the research ethics review is really the potential for exploitation of the underprivileged or of populations. So really thinking about the flow of where the benefits of the research are going to go in your study design is critical. Having a pathway for benefit sharing is really important. Issues around lack of insurance, risk of third-party transmission, and big debates around compensation. That looks very different in different settings. I don't think that the debate should also always be around the high-income, low-income setting, because some of the things are very different depending on whether you have a national health system, all these other things. So context really matters here. And then in terms of having my work cut out for me, these are some of the things that we are thinking of doing to follow up. Finalising the 2 guidance we spoke about. We will be coming back to members of this community to ask you about some of the science associated with the pathogens and pandemic potential, because you cannot disassociate the ethics from the science, or you have bad ethics, as well as you have bad science. Development of associated training materials. I think I have to get cleverer in what I do. Having a set of questions isn't looking like it's working. And then really focusing on socializing our guidance or implementing it. And as you'll see, we have a suite of guidance that we're putting together that includes adaptive platform trials, decentralized trials, and human challenge studies. So we'll be having a regional implementation of those in late 2025. And again, I think we have to update our case studies and add more. And on that, I'm going to finish. And I just want to call out a huge thanks to Susie Bull and Zeb Jamrovitz, because they are the 2 lead authors on the case studies and these 2 documents. And that's it. I'm going to stop. I think I've gone over time.
Thank you, Catherine. That was fantastic. I couldn't stop you. So if you would speak
I'm
more.
on Brazilian time.
Yeah, but we were ready for that. Any questions?
Catherine, do you think there's a false dichotomy or trichotomy between the kind of warp speed vaccine development, warp speed clinical trial development and approval, and the approach you've described there for human challenge trials? Because it feels like the depth of engagement being encouraged, and other elements there take so long that you end up in a situation where the urgent need for your human challenge model maybe has dissipated?
Yeah, so I'm going to answer that in two ways, because I think the governance is fundamentally important here, and then the engagement models. So I think I was making a really strong case that we need to be prepared in terms of our regulatory and ethical governance, right? And if we can do that now in the inter-pandemic period and set ourselves up and have the infrastructure in place, that won't be the thing that's delaying the warp speed, right? So that's my first point. At the moment, I do not think we are well prepared. But I will tell you actually what was really interesting in engaging those 30 national research ethics committee from across the globe is in some senses the Latin American research ethics committees were better prepared than the European research ethics committees because they just had Zika, right? And the European research ethics committees hadn't had any kind of public health emergency for a long time. So I think that's critically important. I think this is a voice that gets missed in these discussions because people are too busy saying, oh, they've got it all wrong, but they're not thinking, how can we get it right? So I think if we do that thinking about the governance of research ethics now, that won't slow it down. But we have to do it, and if we don't do the engagement with research ethics committees, if we don't socialise them, we will slow it down. The engagement of participants, I think, is really tricky and really interesting. We have really good examples during COVID where engagement was done and it didn't slow down the research process. But I'd be really interested to hear from places like Kenya, because I think where you've got these pre-existing community action boards, or you've got a population that's pre-engaged about research, it's much easier to do it. I don't have the answer for how you do a lot of these, but we are working with our unit in WHO and the health emergencies team actually about research literacy, about these types of study designs. Yes, it can slow it down, but I don't think it's incongruous necessarily. But I think instead of just thinking about how we speed up the science in this inter-pandemic period, we have to think about how we do realistic engagement models in the inter-pandemic as well.
Daniela?
Thank you, Catherine. That was wonderful. You mentioned compensation in your talk, and I can please ask a question for you, but also for the audience. My understanding, unless things have changed in Brazil recently, is that we cannot compensate participants financially.
We can compensate for phase 1 studies.
Financially.
I think
Yes.
Lucas, I was talking to Lucas about
It would
this.
be
I think he
really
knows
great
the
to
answer.
hear. Yeah, so that's a very important point because I think, particularly if you think about, team studies and in that context, where obviously they might not benefit from a vaccination that they'll be getting in that particular study. I wonder if you know of other examples around the world where we are in a similar situation where remuneration for time and inconvenience is not allowed in the context of research.
I know that we're doing a review of that, but I
Okay.
haven't seen the preliminary findings. I don't think you're alone in that. And I know that came up in the discussions when they were developing the Indian guidance on challenge studies. But I don't know if Noni and Primus, because I know that they're looking at this in Uganda, Kenya. Who else was at the Mombasa meeting? He was talking about differences in compensation. So I don't think you're alone, and I do think it is an issue. But we are doing a review of it, so I will be able to answer that question in a few months, but I haven't seen the preliminary data.
Thank you.
Okay, Catherine, thank you so much for being here and taking your
That was a
time.
pleasure. Enjoy it.
Our next speaker is someone that I really wanted to come here. When I was in the meeting in Mombasa, in Kenya, and Primus was giving the context of ethical approach in CHIM in Kenya, and then he said, CHIM is not a concept difficult to understand. It doesn't take a lot of formal education to understand it. I said, Primus, you gotta come to Brazil and say it. So, Primus is a social scientist at the Kenyan Medical Research Institute. He currently develops and conducts research with broad focus on ethics-related issues in the context of international health research and in low and middle-income countries. So, please, Primus, come and share your thoughts and knowledge about the theme.
Yeah, hello everyone, and thanks Ethan for the invite to come here and to share what we have been doing in Kenya. Yeah, so I think that's just a brief outline of what I will talk about, and I'm hoping that by the end of my talk, perhaps if there are two things that I want to take home with is the importance of social science in really understanding the ethics of CHIM. And then the second thing also is that I want you to take home, among perhaps many other things, is the role that formal, potential and actual CHIM volunteers can actually play in understanding and even improving the ethics of CHIM as well. So this is just for information purposes. We all know what CHIMs are and the purpose for CHIMs, so I won't spend time on that. And of course, we know they raise a lot of ethical issues. I think with our presenter yesterday, I think raised a lot of them, so I won't also spend time on this. But I think just the issue of deliberate infection, just that itself, it raises a lot of concern across many stakeholders, including the ethics committees. Because we know at face value, one can say it goes against the principle of do no harm. So there are quite a number of things. I think Catherine has also raised some, even in her presentation. And this is just a recap of what the CHIMs that have taken place in Kenya. I think yesterday Rodney did mention this as well. So we've had 6. The first 2 have been completed. The third and the fourth in green are ongoing. And then the last 2, that's the induced blood stage and Shigella ones, should be beginning anytime soon. And the key thing I want to highlight here is that since 2017, we have made a deliberate effort to embed social science and ethics within all our CHIM studies. And perhaps what I mean is that this is just an example. For example, the figure you have there is just the figure of one of the very first CHIMs we did that involves inpatient stay for up to 25 days. So all our CHIMs at the moment, once the volunteers are challenged, they stay in an inpatient facility where they are closely monitored until when they are treated and released out of the facility. So when I talk about embedding, is that even from the time the studies are being designed, we actually discuss with the as well. We try to think of potential issues that could arise and how and what are the type of questions that perhaps we should be exploring together. And as you see there, even from before, at the time of engagement, we actually we are there seeing how engagement is being done. And we actually document like what are the best ways of doing engagement and even consenting, how should we improve that? And even when they are being challenged, we also observe. And we have really learned some really interesting things just by observing people. Like you realize some people, they say heightened state of anxiety at specific times of their residency. And what you learn from that is that perhaps you can even bring in some interventions to improve their participation experience as a whole. See? So even when they are done, they have been released, they have left the study, we do follow-ups. We've had 1 year follow-up studies, like they have left the studies more than a year, and like just engage with them to like get a sense if they still understood what they went through and how the experience was. And so I will just highlight a few of the learnings that we have had from the team over these past 6 years that we've been doing this. So this is more or less, what I had highlighted. We began mostly with the volunteers initially, then over time we expand to also engage lots of a range of other types of stakeholders, like the regulators, like the ethics committees, and even other community stakeholders as well. So these are just some of the outputs we've had from our earlier work, and we've explored a lot of issues in those. We've explored issues around motivations for participation and even decision making. How do people decide whether to take part in these studies? Do they consult others? And who are the types of people they do consult? Whose decisions do they really take more serious than others? We've also looked at issues around just how do different stakeholders perceive what deliberate infection is all about. And interestingly, people do understand these things once you really explain to them. They are not like blank, They do understand them if you are meticulous enough to explain this to them, And we've explored even issues around perceived benefits and burdens of participation, especially with the volunteers. And I will also highlight some few findings on that in the coming slides. We've looked even issues around compensation. It's quite a hot topic as far as this is concerned. What is the role of context even? Because the tendency at times is just to copy what is being done in one context and apply in another context. But what we are learning, like Catherine is mentioning, context is really important. You have to think about your context even on an appropriate levels of compensation. And again, beyond the compensation, what are the ethical issues that all other forms of payment generate, even in such studies? We've looked at those as well. And again, what lessons have we learned, even just from engagement for these years that we have been doing this? What are some key lessons, which Noni will talk a lot about that in her presentation. So in the next 2 slides, I will just highlight just 4 key themes that have been running through a lot of what the studies we have published. The first thing is the issue of payments. And it's easy to run away from this, but the key thing is that payment is an important issue. So researchers need to be aware of that, ethics committees need to be aware of that, regulators need to be aware of that. Payments are important. So many of the volunteers, it's an important issue for them, and you can't take it away from the picture. And again, we find that because lots of the conversations we've had with other stakeholders is that they are concerned that there may be undue inducement. But what we found is that when compensation levels are actually high, people are more, they scrutinize things even more keenly. That's what we have learned from our side. And looking at the safeguards that are even within some of the studies, we actually realize that the risk of undue inducement is really is quite low. Because basically undue inducement happens when you have a very good offer that makes someone to take a decision which is against their best judgment and which could lead to harm. But basically the type of care, like watching these guys in hospitals, like in inpatient, the type of care they have, patients don't even have that care. like they have access to a clinician or physician 24/7, and they basically draw blood every day and to test them. So they actually stop treatments. They bring in treatments when it is really appropriate. So the risk of having really serious adverse events is really minimized. Yeah, and that's something that is good to highlight as well. And we have also even engaged with them, like seeing how have they used the compensation they have received from the studies. And interestingly, it hasn't been it has been good. It has been well received, but it hasn't changed their lives in any way. So these are not, and my idea is that at times we've had researchers and other stakeholders who say, no, this amount is much. But what we are seeing is that that is not the case. It doesn't change people's lives. See? Because the tendency is to reduce it. And once we reduce, what we do is exploitation, rather. See? Then again, on benefits and burdens, we have actually found that volunteers do actually weigh these things. They do weigh the risks, the burdens. They have their own ways of doing it. And at times, it may even be engaging with former participants who have gone through the same studies, just to gauge how things are, how their experiences were, you see. And what we learned is that there are a lot of unusual burdens and benefits that are often not even captured in the ICFs, in the informed consent document. For example, what we've learned is that there is even most of the burdens are actually psychological in nature. But that is hardly mentioned even during the informal consent process. And again, as we'll see, I will show how what we've learned, how we've channeled that into practice. Say. Yeah. Then engagement, I think Noni would talk a lot about that, but engagement is really critical. To have a good engagement platform, it makes a difference, and it really makes the work easier, perhaps for the researchers, and even the volunteers as well. Women participation is also one thing that we see across our findings. And here across the board, there is a strong perception that the way these studies are designed, they disproportionately favor men over women, especially the long inpatient stays. Because women with kids, they struggle. They won't be able to stay in such places for a long time. And again, no care for kids is provided, even by the researchers as well. So in a way, what we are seeing is that most of the volunteers are young men, especially young men who don't have responsibilities like care responsibilities. And for many women who participate, they actually find the compensation really empowering, because it allows them to make some important decisions in their homes, take away some of the burdens from in their homes, and for them it is really an important thing. And most women also find the time in residence a huge relief. It's a time of rest. You can imagine you are home with 4 or 5 kids, then now you're away for 4 weeks. Your food is provided for, your water. So it's more like a holiday for some people. See? And I don't think we should discard such issues. See? So in the next few slides, I will just highlight what is ongoing now. So this is a study that we are just wrapping up. It's funded from the European Commission. So we're looking at the social, ethical, and regulatory issues that are involved with CHIM. So you can get this. This is a protocol that is on Welcome Open, so you can check it out there. But in this study, I think the key thing, we wanted to really carefully document social, ethical, and regulatory issues with CHIM in our setting, and it was a mixed method approach where we do some surveys with participants before they even join the studies. Another unique thing we did was to use a deliberative approach. And I will talk a bit about that as well. And a deliberative approach to consultation is an inclusive approach that you use, and it is based on the understanding that knowledge, the public holds important knowledge about things. So it's not limited just to perhaps to the highly educated or to the scientists and all that. So you could get useful information even from the community, even from participants, especially issues around cultural acceptability of a particular type of practice. So far we engage with 56 stakeholders, which include those from the community, like the community representatives, of field workers, and even former volunteers for these studies. And then importantly, the research ethics committees, the regulatory authorities, the researchers themselves, as well as other former participants. And we explored a wide range of topics over a period of time. So this is just a representation of how this approach looks like. So first we have large workshops where we bring a participant of a particular group together. It's usually for a full day where we just give them information about what CHIMs are. They ask the questions, so we have the researchers there who are able to respond to any question. And after that full information day, then now we invite them in smaller groups for now in-depth small group discussions now, where we explore our questions. And the idea is that everyone is brought on the same level as far as knowledge and information is concerned. And once we've synthesized everything, we bring them to a validation meeting where we actually share what we think we have learned from them, and they confirm that this is actually what had been discussed. So I will just highlight a few things that we have had from that. Compensation is, like I said, is one key thing that we have explored, and what we are finding is that they think that there is a huge agreement that there is need to increase the compensation that is being provided at the moment, mainly because of increased cost of living. The actual amount, there is no agreement on that. And the participants actually understand that compensation is not a wage. It is not a wage, it is a form of just recognition for the time that they have put in a process. And again, there is agreement that compensation should be for CHIM should be the same across the board, irrespective of the type of CHIM. So again, we find that there is the volunteers, former volunteers think beyond the compensation, there should be other ways to recognize their contribution. It could be through certificates or other things that will just recognize what they have put into such studies. Then the topic of risks and harms also is something that we explore with them. And as researchers, we thought these were very complex concepts. But once you educate former volunteers, they can make the difference between risks and harm if you educate them. So, for example, these are some of the issues that they have brought out around perceived risks for the malaria CHIMs we've had, like risk of severe disease, and even adverse events from vaccine or the challenge itself, and again, even fear of health screening results, like screening and discovering a condition that you had but you had no idea about. How do you handle that? So they do have that as well. And we have even looked at harms, which they have also mentioned, like the pain and discomfort, even psychological distress from being away from home, and even loss of property because they are away from home. And we even explore, are there some types of harms or risks that participants should not be exposed to? And again, we find lots of people bringing up issues of the amount of blood should really not be excessive, and the dosage of the parasite should also be reasonable. And again, studies should be designed in a way that it doesn't cause disharmony within the society, within the community. So based on this, this is my last slide. I just highlight a few implications for policy and practice. I think practice a level of our institution and the study teams. For now, when they began, there were concerns around the actual volume of blood that was being drawn out. And what the teams do now is that they actually have tubes that they actually use as part of the engagement and informed consent process to actually show the actual volume of blood that is drawn out. And that has actually improved understanding as well. Then compensation, when they began also, the compensation was like it was done at the end of residency, like a lump sum. But what we are learning is that most of these volunteers are casual workers who need the money on a daily basis. So they have now staggered the payments on a weekly basis. And again, which is really interesting, we have actually found that education is not really, the level of education is not a really critical determining factor for people to understand this. If you are careful to craft out your key messages and all that, people do understand them. And again, Noni and team, they have really helped us a lot on how to craft those messages. Then the lessons we've learned around the benefits and burdens now, the study teams are also including that as part of the informed consent process. And again, we have also learned like pre-submission meetings with the ethics committees, regulators, where you bring all of them together have also been really helpful. Like you bring the ethics committees, the regulators in the same room, and even with formal volunteers, where the regulators have the opportunity to ask some questions even to the former volunteers. And we have found that the regulators and the ethics committees have found that really, really helpful. And another thing I also want to highlight is that as what has made us to be able to deliver more on this is that we have a stand-alone a social science proposal. So we are very proactive. So even while the science guys are still thinking of what to do, we can get things done because we don't really depend so much on what they are doing. Of course, on the policy side, what we've done has fed into the Kenyan regulators guidelines of 2020, then the guidelines that Catherine mentioned. Then I'm also part of, there's an ongoing work on healthy volunteers, ethics of healthy volunteers, which our team is also part of, trying to develop an ethics charter to protect healthy volunteers in clinical trials, which CHIMS are just one of them. And also some members of our team, myself and Melissa, we have been recently selected to join the African Vaccine Regulatory Forum as experts. They want to develop some guidelines also for game on the continent. And again, we are in the process of discussing with the ethics committees in Kenya also to see how what we have learned we can help domesticate them within the guidelines of the country. So I will end here just to thank volunteers, the communities, and even our funders, and again the organizers for the opportunity to share what we have learned so far from our side.
Thank you, Primus. Any questions? Ana, and then Alcino.
I have to say your discussion around payments was very refreshing. We generally don't have, I don't know what the progressive view on payment of volunteers. And I was just curious how that came about. Was it through discussions with volunteers?
Yeah, so we, as an institution, we have a guideline for that, and it was done through consultation with community stakeholders. For example, like you were asking if you have to devote like an hour into an event, how much will it cost, or how much are you ready to request for? If you're able to give like 3 hours, how much would that be? So it involves consultation with a wide range of stakeholders, and we think the voice of the community is really important, because the community actually said there's a level that if it's too high now, we will have a concern with that, because it will raise a lot of suspicion, which we will not be able to address. So the voices of the community and the volunteers, and especially them now, like what we've done with the deliberative process, where we bring in the regulators. They sit in the same room with the former volunteers, the community representatives, and all this. I think it's also helping the regulators think things differently as well. Like now we are in the process of doing some new guidelines on the payments, and yes, so all this, it doesn't just come from, we engage all the stakeholders.
Yeah, I was going to make the same comment as Anna, because here in Brazil, we cannot pay the study participants except for phase 1 or bioequivalent studies. And I really liked this concept that if you do not pay, you are exploiting them.
It is.
And that's a shift of understanding for us in Brazil. Like, they think that this is a voluntary work, and you should do it voluntary and take all the risks. So thank you for bringing this. Alcino.
Primus, thank you. Brilliant talking. How is the ethical system in Kenya? Is there local ethics committee or national ethics committee? And secondly, you cited the WHO guidelines. Have you low or Kenyan guidelines now for the teams?
Okay. Yeah, so with respect to the ethics process, like for our institution, we begin with an IRB at where we are. Then from there, now it goes to a national ethics committee. So once that has been approved, then it moves now to the regulator that that does the review as well. And along the way, I think, like I mentioned, the pre-submission meetings. In the past, it wasn't the case, but like the study teams started doing it, and we have done some interviews. I don't have the results here, but we've done interviews with regulators and ethics committee members who have been part of that process. Because when they were invited, at times, some were like, we think we are a bit conflicted. We can't come and sit with the researchers, and But at the end of the day, what they have really, the way they review is different, because they are reviewing from a point of understanding. Because the question, like, I've been an ethics committee member for many years also, so the question we usually ask, like, what is your responsibility as an ethics committee member? If it's to protect the well-being of the research participant, what do you need to do? You need the knowledge. So if you don't have the knowledge, it means you are actually not being fair to those that you want to protect them. You see? Yeah.
There was insurances after the Kim study?
Insurance?
Insurance, yes.
Yeah, I think.
Or only compensation, money compensation?
No, compensation. Then, if there are any harms, any issues, there's insurance that it covers for that as well.
Mm-hmm.
Yes.
Well, we have to move on because we are passing the Brazilian standard time. So, Oxford is studying ethics. And Alcino was the first Brazilian ethicist to bring CHIM into the discussion here in Brazil. He published a article in a newspaper of wide circulation in Brazil, and that's how I knew I was not alone. So one day I wrote to him just hoping he would answer, and he did. And he brought the bioethicists to the project, which are making a huge difference. So thank you so much, Alcino, and thank you for being here.
Okay, thank you, Professor Elton and Professor Daniela and all the team here that organized this symposium. Well, my talk is about the ethics of controlled human infection models, CHIMs, in the discovery and evaluation of vaccines, treatments, and knowledge for infectious disease. They involve the deliberate infection of fully informed and free volunteer participants with a version of the pathogen causing the disease. Is this ethically acceptable? Should researchers and physicians propose and carry out of such studies? In this communication, after summarizing some aspects against and some aspects in favor of CHIMs, I offer an ethical evaluation of the proposal based on the traditional principles and values commonly used in the ethics of research with human beings, especially here in Brazil. The fact is, in favor of CHIMs, whether for emergency, public health emergencies, or as a new standard for research with human beings in general, there are the presence and combination of the following aspects. Firstly, the prospect of significant social benefits, either with the discovery or improvement of vaccines and treatments in various types and modes of production, platform production, as well as obtain information on pathogens and correlates of protection, usually for serious diseases involving thousands of or millions of persons. In addition to other harms caused to public health or economy or social life. Secondly, the availability of thousands of volunteers whose informed consent can be properly established. Thirdly, the altruistic willingness of these participants to risk their lives and health to promote the greater good. Analogous, for example, to donating a kidney to a relative or a stranger. For the low risk of death, for donating a kidney to save the life of just one person, we have a mortality risk of around 3,400. But in the case of CHIMs in COVID-19, for example, carry out on healthy young people, the information available in one study estimated a risk of 1 in 40,000, or in another study even lower risk, 1 in 120,000. A recent systematic review showed that no deaths occurred in CHIMs studies from 1980 to 2020. Well, but the following aspects weigh against CHIMs in emergencies, such pandemics or endemics, but so as a new standard in risk research. First, the direct risks are of harm or harm to volunteers. Secondly, risks of fostering public distrust in science in general, and vaccines in particular, especially here in Brazil. If death or serious sequela occurs. Third, risks of weakening adherence in current and future researchers and physicians to the well-established standard of not conducting unsafe research with deliberate infection. And 4, doubts about the well-informed nature of the consent of volunteers, with a reasonable suspicion that there is some therapeutic misconception. The deliberate infection of volunteers and the traditional alternatives, phase 1 to 3 of clinical studies, for example, this would be a possible evidence that the principle of non-maleficence, one of the main, perhaps the most important principle of medical ethics, is being violated. Okay, teams and the principles of ethics in clinical research. Considering the normative standard normally used in ethics in research, especially in Brazil, with human beings, a standard that includes the principles of respect for autonomy, non-maleficence, beneficence, and justice, we should recognize, even with some uncertainty, that the aspects the favor, outweigh the aspects contra, against, and lead us to approve teams, or should lead us to approve teams. Why? Because the proper weighting and specification of the 4 principles together, applied to the factual context of many infections, makes the proposal ethically acceptable. Respect for people's autonomy implies recognizing and accepting the well-informed and free will, for example, of the thousands of volunteers available through One Day Sooner, or at least a significant part of them, well, they fully informed and free will to participate and to contribute effectively and altruistically to overcome infections and protecting vulnerable human beings. The proper understand and use of principle of beneficence, which includes, if properly understood, the large-scale benefits to population, the promotion of the greatest good in aggregated terms, for example, in cost benefit, cost effectiveness, and cost utility analysis, also results in being favorable to teams. Especially, for example, this was the case on COVID-19. The principle of justice usually prescribing non-discrimination, but even the fair distribution or equitable distribution of burdens and benefits can also be used clearly in favor of teams. For example, since many people bear heavy burdens and risks in fulfillment of their duties, so that on parity of risks basis, allowing teams is far less problematic than banning them. To these aspects, we must add the novelty of the value of effective altruism. The ideal that includes proposals to do the greatest good by adopting actions with a high impact on generate benefits based on science. In the case of teams, high impact is expected in terms of promotion the greatest good for an immensely large number of people compared to the number of volunteers. Benevolence in this wider sense and the altruism are particular values to especially be included and be recognized in bioethics, at least in Brazilian bioethics, and especially in research ethics. Our local ethics committee and the national ethics committee almost quasi in all the time do not use the principle of beneficence. Very strange. These multiple evaluative aspects related to the specific factors of serious infectious diseases, such as the high rate of infection, lack of varied vaccines and treatments, and insufficient quantity to serve the global population, and the negative social effects of managing these diseases without vaccines or treatments. If we consider the participation only of health people after giving robust informed consent and the special clinical and scientific care in this study solve the problem of the alleged apparent disregard to the principle of non-maleficence, the only one that generates more serious diabetes against chimps. The infringement is merely apparent because empirically the suggested risk benefits weighting seems to fit the ideal of minimum risk and the maximal benefits. We also recommend chimps, for example, if we compare them with risks in other researches, whether in phase 1, the clinical research, where healthy volunteers are at risk when receiving substances still at the beginning of the study, or in research in case of gene therapy. Or if we compare chimps with medical procedures, such as the aforementioned kidney donation, or if you compare chimps with widely accepted social practices that involve the risk of dying or suffering serious sequelae, such as radical sports, sending young people to war in normally obligatory, not in autonomy, or keeping essential and health professionals working in serious epidemics or pandemics, for example. These things involve similar or greater risks and are accepted, and with probably lesser benefits. It can be said that it can also be said that this regard for non-maleficence is apparent because normatively, even if there is deliberate production of risks of harm, or even harms, such death or serious sequelae occurs, it does not constitute harm in the general normative sense of the expression. Or if it does, if it would not be a form of illegitimate promotion of harm from an ethical point of view. Why? Because the free and informed consent to take the risks, together with the care for the volunteers, and the clear provision of greater benefits to society as a whole, cancel out their harmful dimension. In the same way that a terminally ill patients inform the irrational will to die, together with the fact of unwanted suffering, cancels out the harmful of causing death in, for example, assisted suicide or euthanasia. However, if we treat chimps as a harmful practice, we are left with the moral conflict of also causing harm, firstly to the volunteers, since their rational desire to participate will be denied. And denied supposedly in favor of themselves, the volunteers, which constitute a kind of out-of-place paternalism. The volunteers are adult and health person. Secondly, there would be similar harm to the researchers who wish to carry out the chimps. For example, Elton in Brazil. The refusal would be harmful to people suffering from diseases without vaccines and treatments, and society in general. Now, if there is such conflict of moral duties, a kind of moral dilemma, it's not clear that is our duty not to cause the harm, between commas, of doing chimps. It may be that the duty not to disrespect autonomy is also properly required of us and prevails in the choice. Moreover, as we have seen, there are good reasons to accept the balance of reasons tips in favor of chimps, and then the difficulty with non-maleficence disappears, because the interpretation that there is in no way any more plausible. Does the traditional ethics of research with human beings indicates that chimps are ethical? And if there are altruistic, full informed volunteers and serious researchers interested in them, it is unethical to prohibit them and not to approve them. Conclusion, it follows from the above assessment that research with chimps, at least in scientifically and ethically designs, is justified and should be recommended and approved. Ethics committees, whose members normally consider the 4 principles of ethics, the biomedical ethics that we have used here in Brazil, but who also have time, information, and researches to apply moral reasoning appropriately, would probably come to the same conclusion, or should to do so. And all of us in general, as citizens, should also conclude this way and support chimps. Thank you.
Thank you, Alcino. We are very advanced in time, so we may have time for one quick question. So if not, let's move on. Thank you so much, Alcino.
Okay, thank
I'm
you.
sorry. Now, our next speaker is Brunello Stancioli. Brunello holds a degree in law, a master's and doctorate from Federal University of Minas Gerais. And he also has been at Oxford University in the Faculty of Philosophy, studying CHIM. One of the subjects was CHIM ethics. And he is one of my team members and is involved in ethical justification for CHIM in Brazil. So thank you so much, Brunello, for being here and to talk with us about it.
Hello. First of all, I would like to say thank you very much to Professor Helton. Thanks for having me here. He knows how important this is to me. Okay. The second point is that there was a Brazilian composer, Tom Jobim, very famous. He has written The Girl from Ipanema, and he said, Brazil is not for beginners. So our situation is very complicated here. We haven't one human challenge project. I think no one. And supposedly there is no problem dealing with this. Well, I'll start with the very same 4 principles in bioethics: the beneficence, the maleficence, the justice, and the autonomy. Brazil has these principles in all its legislation. All our legislation is pervaded by those principles, from the Constitution to presidential act. The problem is that how we read those principles. Because if Greece gave us logos, the Iberics, mainly Portugal, gave us charity and paternalism. Our hermeneutic of the 4 principles is extremely paternalistic. And the apex of the paternalism is just put away the autonomy and the liberty of the subject of research. So let's present here a general model of scientific method. We have the subject and the object. The subject acts, modifies, makes measurements, and the object is just manipulated. If you translate this to a medical relationship, you have the physician and the patient. The physician has all the knowledge and the patient is manipulated. Or we can have the researcher and the subject of research in this imbalanced relationship. So we must equalize this in order to give them the very same opportunity to talk, to defend, and to understand the problems. Both of them must be considered persons, not human subjects. They must be persons. And the word person is very, very complicated and it has a historical meaning that is fantastic. But I just say that person comes from Latin personare. It's a mask. It's a mask, and the sounds go through the mask. So it's a hole in a theater. You present yourself in the society. There is a Brazilian philosopher, Professor Enrique Vaz, and he said that persons only exist in plural. And the word person as an actor show us that we act publicly in every stance. So there is no person without another person. Even grammatics, you have the first person, the second person, the third person. Even in Trinity, Christian Trinity, we have Father, the Son, and the Holy Spirit. Well, how can we think to balance this situation, as I've said, of researcher and subject of research? They must be considered persons. What does it mean to be persons besides being a mask? Persons implies that the individual has his own sense of identity. There are many ways to describe identity, from John Locke, Mary Chestman, but I'll take the idea that identity is the sense of belonging to a community. He has his own values. He has his own taboos. He has his idea of good life. So, when it enters in a conversation with the researcher, both must be committed to mutual understanding, reasoning and understanding in these positions. And the autonomy appears in the moment of consensus. So autonomy is not a characteristic of the subject of research or the researcher. It emerges from this relationship. So in autonomy, we have an agreement of what is permitted, what was not permitted in our relationship. In Brazil, we tend to prohibit, not allow, every kind of benefit for the subject of research. We cannot pay. You can pay just a bologna sandwich after the research, something like this. And it tends to patronize the subject of research. If you are poor, you are vulnerable. Well, poor people have marriage, have kids, have education, have values. As I've said, they are persons. So, in this dialogue between two persons emerges the autonomy, as I said, as a byproduct of this conversation. Okay? So, if I have autonomy, I can decide whatever I want in this relationship. I can ask for specific treatment in some cases, and I can even renounce to some values in the beneficence principle. If you're committed to a noble cause, for instance, you don't want to have compensation, monetary compensation, etc. But in Brazil, if you have this situation occurring, the research will be prohibited. We tried to make the very first human challenge in Brazil, coordinated by Professor Elton, with the infection with worms. Amarelão, how can I say this in English? Ancystomiasis, yes. And inject this in the subject of research and try to develop a vaccine, the very first vaccine against worms. But first, the decision of the local committee is that the subject of research were vulnerable. Why? Because they are poor? Well, in a second try, we got another no in our face, but now saying that the patient didn't have benefits with the research. It seems a little bit contradictory, isn't it? But, in fact, defending the ethical point of view, I think if both are together, both are trying to get an understanding, a mutual understanding against that, we have the emergence of autonomy, and I can say you, autonomy only exists if there is solidarity. Thank you very much.
Thank you, Brunello. We may have time for some questions. Anna.
I don't know if it's a question so much as a comment, and maybe Catherine or somebody else can comment on my comment, but I thought it very interesting, your reference to person, and whether or not, in all of our protocols, our consents, we always refer to people as research subjects or research volunteers as opposed to research persons. And, and it does change sort of how you perceive that, and I wonder if that's something we should think about.
Yes, that's the idea. I think so. Person is much more important than subject of research.
Anna, that's the thing that emerges when you are talking to people from a totally different area. Like, we are physicians, we have one perspective. So we bring a philosopher and a lawyer, it's a different perspective.
I did not present myself. I'm a law professor in the Federal University of Minas Gerais, but I can assure you, I'm a nice guy. Thank you.
Alcino, and then, you want to, okay.
Anna, I think you raise a really interesting point. Interestingly, the subject comes straight out of the American NIA. It's come out of, and that's where the dominance in the past has come from. And I think there is a movement away from using subject, I think for exactly the same, for some of the reasons you are very well articulating, that it delineates, language is important, and subject and participant, there's a power imbalance that you are increasing by using that language. But getting away from that, I think, is truly tricky because it's embedded in all our regulations and legislation and the rest of it. But I think going forward, and even the use of the word, and I deliberately said, when you said vulnerable, I think so really, it denotes protectionism. So thinking about different uses of language, I think is really important. But I want to come back to, I loved your comment about Brazil's not for beginners. And I think I come from a country that is for beginners, Australia. How do you square the circle on this? Where is this paternalism coming from in Brazil? And if you're thinking, and this is not just about challenge studies and your relationship with the governance mechanisms, whether it be regulatory mechanisms or ethics governance, how do you start to shift that really paternalistic view other than changing the language? Because I think that's a critical underpinning of what we're talking about here.
Thank you very much. Well, I propose this change of words because person has a long history and a strong philosophy connected to it. But I would say that our paternalism comes from the Portuguese. Sorry if there is some Portuguese here. But it's the problems that the Portuguese were very Catholic, and our state just became separated from the church in the end of the 19th century. So you have this idea that we must be conducted by someone, to have a shepherd for us, so they can overprotect the subject of research even against his will. That's the big problem, okay? We must protect the vulnerable, of course, but we cannot just discharge his idea of autonomy and good life and why he is in that research. As I said, it can be a noble cause, for instance. Thank you.
Okay. Thank you so much. I think that was very rich, very different from what we are used to. So thank you, Brunello. So we have our lunch break now, and we will we'll resume at 2 sharp because we are right now Brazilian time.